All right. So yeah, welcome everybody. So this is one of the webinars that's organized by the Machine Learning and Systems and Computational Biology COSI of ISCB. So it's my great pleasure to introduce to you today Jianlin Chen from the University of Missouri, where he is director of the Bioinformatics and Machine Learning Lab, and also the William and Nancy Thompson Missouri Distinguished Professor, which I'm not sure exactly what it is, but it sounds really cool. So I'm really happy to introduce Jianlin today because I've known him since 2005 when we were both PhD students in the same lab at UC Irvine with Pierre Baldi. We were working on entirely different things. I mean, machine learning and bioinformatics, but different topics. And Jianlin was already working on machine learning for protein structure prediction. So the 1D, 2D, and also 3D levels. So if I recall correctly, there was both recursive neural networks and SVM involved. So neural networks were not so fashionable at the time, SVMs were. But that's still one of his areas of interest, protein structure prediction, and also neural networks. So I think that's the topic of today's talk. I also wanted to mention that, I mean, since 2006, when he graduated, got his PhD, Jianlin has got numerous awards. One of them is an NSF Career Award in 2012. And yeah, I think that's all I wanted to say to introduce today's talk.
Okay, thank you very much for the nice introduction. Should I start to share the screen, then start my presentation?
I think so. I mean, Sayani is making the rules, but...
Okay.
Yeah.
Okay.
I mean, anything works. I think maybe you can introduce and then, yeah, go ahead with the slides.
Okay. Can you see my screen?
Yes.
Yes, I can see it. Yep.
Great. Thank you very much for the, Chloe. Thank you very much for the nice invitation and a nice introduction. It's my great pleasure to introduce present my recent research on protein structure prediction today. So this work is based on one of our paper published in the Informatics Journal on protein content prediction, and also our research results in the last worldwide competition, in the protein structure competition called COSI-13, and also our latest advance in this field. I will first give an introduction to the protein structure prediction problem and the protein content prediction and the distance prediction and why they are very important. And then I describe our deep learning networks methods for protein contact and distance prediction. And then I describe how these distance and contact prediction can be used to build the three-dimensional structures of proteins. And then finally, I will conclude my talk with a few observations. So we know that protein structure, protein is a sequence of amino acids. And this sequence needs to fold into three-dimensional structure in order to carry out all kinds of biological function in cells, like catalyzed chemical reactions. So knowing the protein structure is very important for us to understand the disease, because many diseases are caused by the mutations on the proteins that are disrupting the protein structure. And also design new proteins. For instance, design a new enzyme that can convert the biomass into energy more efficiently. And also precision medicine. So if we know the protein structures and how mutation changes structures, we can find the better treatments or drugs to deal with these mutations. And also it's very important for drug design. This is particularly relevant today because all of us are really going through the painful coronavirus pandemic, right? So for instance, here is several proteins of the coronavirus. particularly the structure of the spike protein, this one, that actually used by the coronavirus to enter into the human cells. So if we know this structure, then we actually can find the drugs that may able to bind to this structure, then inhibit its function. So this actually structure-based drug design is very useful for quickly reduce the time to find the effective drugs. So it's very important. And currently most protein structures are determined by experimental techniques like X-ray crystallography, NMR, but these techniques are very expensive, so they cannot be applied to all the proteins. That's why we need to develop computational methods to predict protein structure. So recently, actually, we are applying our methods to predict the structure of the COVID-19 proteins. So we actually work with COSI Commons. This is a community-wide effort to predict the structure of the COVID-19 proteins and also shared with community. And we are participating in this effort organized by the COSI. So we know that actually artificial intelligence is revolutionizing many fields, right? Like image processing, voice recognition, self-driving cars. And we also know that AlphaGo, so I mean, everybody knows Google's AlphaGo, actually is an artificial intelligence, machine learning, deep learning driven Go player that actually beat the worldwide champions Korean player. So a couple years ago, that actually makes the machine learning, deep learning, and artificial intelligence very popular. Actually, you probably also heard of the Google's another application. It's called AlphaFold, that are developed by Google to actually fold the proteins. Okay, actually they did very well in the last worldwide competition of the protein structure prediction. That's called the COSI-13. So actually, because they are very successful, so they are greatly advertise their success. So a couple of years ago, there's so many news on the internet about the AlphaFold. It's called Google's DeepMind ACEs protein folding. Another one is DeepMind's Chris playing games with AI after protein stakes with machine learning code. And Google's DeepMind predicts 3D shapes of proteins. So this actually demonstrated that machine learning, artificial intelligence actually is able to make great advance in a major scientific problem that is protein folding. So indeed, Google did very well. Their methods were ranked #1 in the worldwide COSI-13 competition of the protein folding. And our method actually also did very well. Our method was ranked #3. So I also like to point out that AlphaFold, Google is a newcomer to this field. So AlphaFold is great, and also is built on the original ideas and strategies developed by the protein folding community over the many years. The particular advances in the deep learning and the contact distance prediction. So that's actually drive the whole field, revolutionize the whole field. That's what I'm going to talk about today. So we know that protein structure prediction is to use computer to produce the three-dimensional structures from protein sequence. So there are two different ways to do protein structure prediction. One is actually very simple. It's called a template-based modeling. It's pretty much like the nearest labor machine learning method in the machine learning. So basically, I have a target sequence. I don't know its structure, but I know some structure in the protein data bank whose structure is known, and the sequence is similar to this target sequence. Because they have similar sequence, they tend to have similar structure. So I can search this sequence against the data bank, find the identical similar sequences, then build a sequence alignment. Then based on that alignment, I can transfer the structure, the known structure called a template, to my target to build a structure for my target. So this is template-based modeling. So basically, find the nearest labor, then use that as a template to build a structure. It's simple. However, the problem is for many proteins, we cannot really find any good templates. So then we need to build a structure from scratch. That is called AB initial protein structure prediction, or called template-free protein structure modeling. Actually, it's considered one of the top 125 unsolved scientific problems by Science magazine. So it's a major challenge our community, bioinformatics community is facing. Now, people have studied this problem for many years. The first kind of method is called a physics-based modeling. So basically is a protein sequence can fold into many different shapes, but each shape has a different kind of free energy. The native structure, the true structure, tends to have the lowest free energy. So then I can sample many different structures, then try to find the structure with the lowest energy. So the challenge is the energy is very complicated, involving thousands, hundreds of thousands of atoms. So it's very hard to accurately calculate the energy, and it's very hard to accurately sample all the structures. So then later in 1990s, another method was proposed. It's called fragment assembly. So basically is you divide a protein sequence into smaller fragments. So you can get a structure from smaller fragments from the database, then try to assemble them together. This approach, represented by the Rosetta, actually can fold some smaller proteins sometimes well, but they cannot deal with larger proteins. So still, fragment assembly cannot consistently build the good structures even for smaller proteins. So it's a major advance, it's not sufficient. Now, the most popular method now, actually that revolutionized the field now, is actually the contact or distance-based modeling. So idea is simple. So basically give a sequence. I first use machine learning to predict the context between amino acids or the distance between the amino acids. I get a 2D contact map or distance map. Then I use this as guidance to build a three-dimensional structure. Basically, I try to bring the residues that are in contact closer, the residues are not in contact further away. In that way, I can build a three-dimensional structure. And here I like to use one example to illustrate the idea of the contact map. So here is, suppose you have a tertiary structure. So I have one residue here, I have another residue here. Because I know their coordinates, so I can calculate the distance. If the distance is lower than the threshold, let's say 8 angstrom, I say they're in contact. So I can actually represent it as a dot in the 2D contact map, right? So position I and J will be a dot contact. If they're not in contact, then it's a blank, right? So in this way, I can have a 2D representation of the three-dimensional structure. So here I'd like to show you how this 2D representation actually captures the patterns in the 3D structure as well. So here's a protein that's starting from here. It's a beta strand, then have another beta strand 2, then for the helices, the beta strand 3, then beta strand 4. So that's one simple fold. You can see beta strand 1 and 2 form some context between the residues in the two strands. That's corresponding to these context, this segment perpendicular to the diagonal. So then helices also, residues in the helices form some context in the local context. That's a context along this band, along this band. And also then you have strand 3 and 4 forms context here. I need to use the, so forming the context here. And then I have another strand 4 and 4, 1 and 4 that forms the context here. Long range context, because the strand 4 are very far away from on the sequences. So, and more importantly, once I get these contexts, if they're accurate enough, I actually can reconstruct the three-dimensional structure, right? So then I find a way to build a three-dimensional structure by first predicting the contact map. Then the key question is, give a sequence we want to predict whether or not two amino acids I and J, any two amino acids I and J in the protein sequence are in contact. That means their distance is below a threshold. So this is a classic contact prediction problem. And this problem was first proposed in 1994. So, but it didn't receive much attention until 2010. Even many methods have been developed, including the methods we developed. I developed with Dr. Boddy, my former advisor, and when I worked with Chloe in 2005. So a lot of nice methods, machine learning methods, neural networks, and support vector machines, but they didn't receive a lot of their attention because the accuracy was not accurate enough to fold the protein structures consistent. Then two major advances in the field make these more and more powerful. That is co-evolution technology, invented in 2011, and also the deep learning technique introduced to this problem in 2012. Those are the two major advances drive this field forward. Then later, contact prediction has further demonstrated improved the ab initio structure prediction in the COSP11 and the COSP12 protein folding competition. And later in the 2013, that's 2018, Google's AlphaFold, our method Multicon, RapidX, DMP-Fold, and ITaser several methods perform extremely well, clearly demonstrated that contact and distance prediction can fold many protein structures, even very complicated topologies well. That clearly demonstrated this is a route to solve the problem. So now I'd like to describe these two major advances. The first one is called convolutional analysis. So basically is I have protein sequence, right? It folds into the three-dimensional structure. So here I have residue 1 and 2 are in contact. Because they are in contact, then they have some physical restraints to maintain, to make sure they are maintaining the contact. For instance, if this is charged one, other one need to be negative charged, so to maintain the contact. If the one is mutated to hydrophobic, other one has to also be mutated to hydrophobic to maintain the contact. So basically, they have the correlating mutations. And these correlating mutations will be reflected in their multiple sequence alignment. I get a sequence, I search all the related sequences, I build a multiple sequence alignment. So you can check the position I and J. If they are in contact, if one residue is changed, other residue has been changed accordingly to maintain the complementarity, right? So then Marx and others figure out a way to calculate the direct correlation between the two columns positions in the multiple sequence alignment that are caused by the co-evolution. And it need to be direct rather than indirect. So this, if you have more sequence, this you're going to have stronger signals to integrate the co-evolution. And this can also be used as a feature for the neural networks methods to predict the context. So that's a first major advance. Another major advance is on the technology side. That is the deep learning method. So in 2012, our groups and other groups introduced these deep learning techniques to predict the context. So here's one deep learning network to predict context. We have take the information about window around one amino acid and another around another amino acid, use the input to use deep network to predict whether or not the two residues are in contact. And this advanced the field. And this technique was introduced by informatics field at the same time as deep learning getting popular in the image processing. So here's the ImageNet competition. So 2010, 11, they used shared neural networks. And in 2012, they start to use deep networks, then continue to use it, drastically improve the accuracy. Pretty much like what we have seen in contact and the distance field, improvement in the contact distance prediction field recently. So however, this method still has a problem because we treat each pair of amino acids independently. However, we know that contexts actually are clustered together. They have correlation. So actually we need to use even better technology to capture the correlation between the contexts. So the earliest attempt to do so is using the 2D recursive neural network. So you basically use the input information between all pairs of amino acids as the input stored in the matrices. Then use a 2D recurrent neural network to predict the entire content map. But at that time, this method, we haven't used a deep network. The computation power is not strong enough to get the high accuracy at that time. Now today, I'm going to actually introduce our recent attempt to address this problem, use the deep convolution neural networks to predict the context and improve the accuracy. So convolution network is a powerful technology that first achieved success in the image processing. So I use one image processing example to describe its basic concept. So here I have image. I want to classify into many different categories. So idea is I first see the pixels. Then from these pixels, I take a window of the pixel, generate a higher level feature, right? Could be the line segments. Then these higher level features, I take a window around these higher level features to generate even higher level features. I continue to do this convolution process. My feature get higher and higher levels. And then it's sufficient, contains sufficient information, then I can make a final classification. So the power of this convolution is, basically is, it can automatically attract features without handcrafting, generate handcrafting features. So that's one. Second is feature composition. So lower level features are composed into higher level features continuously, then your feature become global and global. So this is very powerful technique. Now I'd like to show you one example to see how the convolution works. So basically you have input matrix, convolution, basically is a weight matrix. Take a window input, small window input times the weights, generate a feature, right? So then in this way, you can generate a feature map. You can continue to do this from layer to layer. So that is how convolution works. Now I'm going to demonstrate how we can apply this technique to predict the content map, because content map pretty much like an image, right? So our input actually will be a sequence, a series of matrices, right? The 2D matrices. The dimension is L by L. L is the sequence length. So each matrix may store some information, like convolution. I can calculate the convolution from the multiple sequence alignment of this protein, then calculate the convolution between any two pairs amino acids stored in a matrix. I also can use other relevant information for any pair of amino acids, like a secondary structure, shortness, stability. I store all of them into different matrices, pull them together, I get an input volume with multiple channels, right? Then I use a window, take a small window, take one small window around each pair of amino acid to generate a feature. Then I can slide this window to generate the entire feature map of the same size as the input feature. So then I get a sequence, a list of the feature maps. Then this feature map can be further used as input for the next convolution layer to generate the next round of the feature maps. I continue to do so. My feature get global and global. The reason is why is like this. Look at this feature, right? Look at this feature. This feature is generated from this square window of the input, right? Each of the element in this window is coming from one small window in the previous layer. So this window is generated from a larger window in the original input window here, larger than the window generated this feature. So the feature in this layer is more global than the feature in this layer, right? So I continue to do this, my feature become global and global. And finally, it's global enough, I can use the window around each pair in my final feature map to predict whether or not two amino acids are in contact. So I get a contact map. So that's a basic architecture we proposed. And we can use this architecture to build a more complicated architecture. So here's the idea. Here's the input information about a protein. Then I use these multiple basic convolution networks I just described to generate contact prediction at a different distance threshold, okay? Then all these contact predictions together with original input can be pulled together as input to another convolution network I just described to generate my final contact map at a particular threshold, let's say 8 angstrom. So basically this architecture actually predicts the distribution of the contact map because at a multiple different threshold, you get the distribution. So we trained this network on 1,426 proteins with known contact maps. Then we validated on the 196 proteins. Then we blind test them on the COSPA 10, 11, 12 data sets. So here I'd like to show you why this technique works well. The first key advantage is it uses global information, right? Because I use the input information of the entire, all the pairs in the protein as the input. I generate the entire contact map. So the correlation between the contacts can be captured in the learning process. And also I use the global information because I use the water input information. They have been captured by the convolution process. Now I'd like to show you the one example to demonstrate how this actually improves accuracy. So on the left is a contact map predicted by the convolution method unknown, or the CCM PID. So the red dots represent the predicted context. The blue dots represent the true context. You see the predicted context overlap somewhat with the true context, but there's some noise here, right? There's some noise. Using DNCON2 convolution network method, you get a predicted contact maps that noise has been removed, right? You see noise has been removed because they don't have strong correlation with other contexts. They are removed, largely removed. And some missing contexts also have been filled in. So it's get a denser here, right? So for this case, the DNCON2 contact prediction, convolution-based contact prediction drastically improves the accuracy of the top number of the predicted context, you can see. So we test on the COSPA 10 to 12 data sets. So here's for different data sets. That's the best measures in each COSP experiment when the blind test in the COSP experiment. So you can see DNCON2 concept, Raptor X, of the best methods in those three COSP competition. Here's a popular method called metacycle. That's accuracy for the metacycle. For the top L by 5, L is less, contact predictions, accuracy. And the DNCON2, you see the accuracy is higher than all of them in that on that benchmark. Now, that's the results we publish it. And then since we publish it, we further try to improve the contact predictions. We move into 2 directions. The first is we try to classify distance between residues into multiple distance intervals directly using multi-classification, right? So that actually has been done by the RapidX AlphaFold and DMP fold in the COSP 13. Instead of use multiple binary contact predictors, we directly use multi-classification. We also want to further in another direction is we try to directly predict the real value distance between any 2 amino acids. Not a classification anymore, just predict the real distance. So basically is we give the sequence, we try to predict the distance map, then use the distance map to build the 3-dimensional structures like this. So here's the more advanced architecture we recently developed. That has been published in the bioRxiv. So basically we have input layer, we have normalization layer, convolution layer, max out layer, then we have residual block. This residual block repeated many, many times, 16 times actually. So each residual block has the normalization, convolution, and a squeezed block layer. And then finally, we have last convolution layer. This convolution layer is used, output of this convolution layer is used to predict both multi-classification distance map and also real value regression-based distance map. So basically it's also a multitasking. So we try to do the two things using the same architecture. So here's the results we evaluate on the COSP hard targets. So for the top L by 5 contact prediction, here's a precision. So you can see the deep dist R method using multi-classification has the highest accuracy, 0.79. Second is the deep dist use real value. Then you have the rapid X, then you have AlphaFold. The similar trend you can see for top L by 2 contact predictions or top L contact predictions. Basically, these methods outperform those methods on the COSP13 dataset. We also compare the real value distance map prediction and also multi-classification distance map prediction. So you can see based on the top L by 5-2 or L contacts, the multi-classification performs slightly better than that. However, if you use the root mean square error between the predicted distance and the real distance, deep dist real value distance prediction actually is slightly better than multi-classification. And Pearson correlation, they are similar. So basically the two methods looks like are complementary. Both can achieve pretty good results. Then I like to describe how now we can use this contact or distance map to build a 3-dimensional structure. So basically I get a sequence, I get the contact map between them, I like to build the 3-dimensional structure, right? So idea actually is very simple. For instance, 3 points can form a triangle. If I know the distance between them, I can uniquely define one triangle, right? So basically here, I just have more points from my protein sequences. But if I know the sufficient number of the distance between them, I can build a good 3-dimensional structure. Now, here's a pipeline, one of our pipelines to do these 3D structure modeling from context called convolutional. So basically, from sequence, I can predict the second structure and more importantly, the contact map of distance map. So these contacts can be converted into the distance restraints between amino acids and atoms. Second structure can also be converted into dihedral angle restraints, hydrogen bond restraints. All these restraints will be used by the distance geometry to build a 3-dimensional structure. So we get some sort of structure, but it's not ideal. You see the second beta sheets are not well formed. Then we go to second stage of modeling. We try to detect the beta strands that may form beta sheet, then add some actual restraints to make sure they can form some good hydrogen bonds. Then finally, we build another structure that actually have the nice beta sheet. So the key feature of this approach is contact distance play a dominant role in modeling, basically drive the formation of the tertiary structure. That is different from fragment assembly. That is use distance to select the conformations generated by simulation. So the good thing about this method is they can build complicated structures well if there's a sufficient number of the accurate distance. Otherwise, it may fail. Okay. We test this. Now I like to use a cartoon to demonstrate this idea, right? So here you give unfolded structure. I try to fold them to satisfy the distance between amino acids. So these shape represent how well they satisfy the distance or called energy, right? So if you satisfy distance well, then you have the lower distance error or lower energy. So then you get a good structure, right? Here I'd like to show you one example, COSP12 example. We made a good contact prediction and also good structure modeling. So here is contact map. Blue represents the true contact map. Red represents the predict top L by 5, 52 predicted contacts. They're symmetric, so I only show the upper triangle, right? So you can see the predictions match with the true context well. The precision actually is very high, 96%. Coverage, the recall is 21 because we only select 52 contexts. Then I try to visualize this context, use these virtual line segments on the structure. You can see this context actually captures the interactions between the residues within that beta sheet, between the beta strands well. And then, not surprisingly, if I use this context to build a structure using confold, I can superpose the predicted structure, that's the red one, with the true structure, that's blue one. You can see they superpose well. The MSD of the core area is actually 2.3 angstrom. That means actually it's a pretty good prediction. Here's another example we got in the blind COSP13 protein folding, worldwide protein folding competition. That's one I'm pretty proud of, okay? So here is the real structure. We know later, after we made the prediction. Here's the context map. Blue represents the true context maps. The red represents the predicted context. So you can see the accuracy is pretty good. Top L by 5 is 100 accurate. Then we use this context to build a three-dimensional structure. I superpose the predicted structure purple with the true structure. You see they superpose very well for this protein with a complicated topology. TM score is 0.8. That means it's a high quality prediction. You also can see the context of this predicted structure overlap with the context with the true structure. So all overlap pretty well. That means contexts are well implemented. However, if we use the context as input for a fragment assembly, a version of the Rosetta, you see the structure cannot fold very well. So, and also the context in the predicted structure actually do not overlap well with the true context. So this demonstrated that this kind of distance-based driven approach can actually solve the complicated protein structure well if you have good distance or context prediction. And actually in the COSP13, we made good predictions for a number of the hard targets. You can see all listed here. We predict the correct topologies for these proteins well. So that actually is had never happened in our previous participation in the COSP experiments. So that's why we consider a breakthrough. Now, recently we applied the distance prediction we generated by our deep dist real value distance prediction and also multi-classification distance prediction on the COSP13 hard targets. You can see the TM score of the top model built from distance, 0.487. It's higher than DMP fold and also our early methods confold 2. So if we select the best of the top 5 predicted models, actually its average score is 0.52 because the 0.55 means the topology is correct. 0.52 means actually it's on average is good prediction. And for 23 proteins, we get the predicted out of the, we predict the correct structure as well. So of course, we are still continue to work on these. Currently we are preparing our methods for the upcoming COSP14 worldwide protein structure prediction competition. So that will start, I think it's the middle May next month. So we're looking forward to this competition. Finally, I would like to conclude my presentation with a few observations. First is context distance prediction actually is a key direction for solving the protein structure prediction problem. And machine learning, particularly deep learning, is a key technology. It's going to advance context distance prediction. And we believe that protein folding prediction problem may be largely solved in foreseeable future. So we can see the drastic advances in the field driven by the deep learning. I think it's considered a major success, will be considered a major success of artificial intelligence, deep learning solving a major scientific problem. And deep learning will continue to play a major role in the drug discovery, in studying the protein-protein interaction. So there's still a lot of work for our bioinformatics community to work on. So finally, I'd like to thank my students who are doing this work or did this work. So my current students, so most of them actually are working with me now, preparing for the upcoming COS14 protein folding competition. And also I'd like to thank my former PhD students who made a significant contribution to this project. I also would like to thank National Science Foundation and National Institute of Health for supporting this research. And thank you very much for your invitation and also attention.
Thank you very much, Dr. Cheng. It was a very insightful presentation. So any questions for our speaker?
For our attendees who are new to Zoom, if you hit Control-H on your keyboards, you will bring up a chat window and you'll be able to type in any questions you have there.
Should I stop the screen sharing?
I think I have some questions from the from the presentation. So in the DNCON2, you have used in the second section, you have used 8 Armstrong. Was there any specific criteria for using that?
Yeah, that's a great question. That's really a very insightful question. That basically just a common threshold used in the community. In early days of the competition, people try to do the binary competition. You need to choose a threshold. Then people figure out that 8 is a reasonable threshold. However, it's not there. In many cases, 8 Armstrong threshold cannot actually capture all the patterns in the protein structure. So then people try different thresholds, 8 Armstrong, 12 Armstrong. Then we evolve into multi-classification of the context and also the real value distance prediction of context. So it's just like early days, it was convenience. People choose 8 Armstrong, then adopted by the CASP protein folding competition. Then most people started working on these. As the technology getting better and better, then we actually can predict the distance in greater details at multiple different thresholds and even real value. And this actually drastically improves the performance of the 3D modeling if you know the distance in greater details. So that's a great question. The field should has respond to. Yeah.
Any questions?
I actually have a question. I guess shall I ask it directly or?
Sure.
I was wondering, yeah. Jianlin, if you could comment on the computational resources that are required. Like, do you need any, I mean, once you have trained your model, do you need any specific architecture in order to run it for one protein, and how much resources does it use?
Okay, that's also a great question. So resource has been always a challenging issue for training the deep learning model. For this context prediction, so basically we input is a matrix. We have many dimensions, channels. So it requires, so we have larger data set. Let's say you have 10,000 proteins in your training data set. So then you need to predict the context map. So it requires a lot of the memory. So we need to have the better GPUs. That's why in early days, we are using the old NVIDIA GPUs. We couldn't train on very large data set. We only can train several thousands of proteins. Now we have several better GPUs like the NVIDIA's V100. Then we have larger memory. We can load more proteins into memory. We can train it faster. So we be able to train our deep architecture on, let's say, 12,000 proteins also. And in a reasonable amount of time, let's say several days or 1 week, something. If you have even more computing infrastructure, then you actually can train on much larger data sets because there are many more proteins in the protein data bank that we can leverage. And also you can train on deeper architecture, because deeper architecture, currently we have around 100 layers. Some people in the field have tried. I think the Google AlphaFold, Google team, the DeepMind team, they have much more computing power. I think they have trained, in the last class, they trained under more than 200 layers. And I believe you can go even deeper. As you get more and more data, you can go deeper, you get a further performance improvement. So computing infrastructure certainly is a very important thing for developing such a predictor for the contact distance prediction. So as we get a better computing architecture, I believe we're going to have better architecture, deeper architecture. We use more data, we're going to get a better performance. Yeah, that's a great question.
Thank you.
Dr. Cheng, so there's one question on the chat that is by one of the participants, that is, to which extent did you use available PDB structures to train your method? How many of them, and what were your choice criteria?
Okay, that's a great question. So in the current PDB, there are around 160,000 proteins, right? 160,000 around that. That looks like a lot. However, many of them are actually similar to each other. So then basically redundant, particularly some big protein families, they have many sequences, they're redundant. So it's important to remove the redundancy. Otherwise, these proteins will be overrepresented. They're going to make your deep network biased toward some special families. So we basically reduce their data set by reduce proteins that have high similarity with each other. Basically, use threshold is common threshold used in the field is 30% of sequence identity, pairwise sequence identity, or 25%. If it's higher than that, so will be removed from the data set. So there are several good data sets. The one we use in the DNCON2, we have several thousand of them. Then we have DNPfold, that's a pretty good one. It's larger than that. We also have ReptX data set. So now we actually create one from the entire PDB. After sequence reduction, we get around 1,200 proteins. We also select based on the quantity of the structure. Sometimes we remove the structures that have the very low resolution. So that also is important to get a high quality, highly accurate training labels, contact maps. So yeah, basically our experience is that using more data tend to improve your performance. So of course, you need to have the better computing infrastructure to support using more data to train such a network. And also we also use the Google TensorFlow. So it's a very nice deep learning package. So make it easy for us to try different architectures, implement them quickly to test them. So I encourage all of you guys using the package like the Google TensorFlow, Signal, or some other PyTorch architectures to deal with this data that drastically improve reduce the time you need to implement those architectures you try to implement.
Okay, thank you very much. So any other questions? Okay, so I have one last question, and if there are no more questions, we may be concluding the session after that. So I saw that you have put up capital L in your figures, like that length, capital L. So, so what does that represent?
Okay, yeah, that's a great question. I'd like to go back to the slides such that we can explain it more clearly. So I think it's here. Yeah, here. L is the sequence length of the protein. So because it's basically the number of amino acids in a protein. So because we need to get the information between any two amino acids, so then you get a L by L input matrix to capture the convolution between the any two amino acids, I and J, and also secondary structure and many other information you think useful. So you get a multi-channel input volume, image volume. That's at the input. Yeah. Yeah.
So
Output
the
also
question,
is L by L. Yeah.
Because the thing is, then I was looking into that the observations. So you were showing that the accuracy and the precision. So there, I guess, I mean the L what was the L value for your cases? The range.
Yes. Okay, that was a great question. So when we calculate the accuracy, because we predict the we predict the probability of context for any pair of amino acids, right? So you have many pairs. Basically, it's L times L divided by 2. So it's symmetric. L times L minus 1 divided by 2. So that's the total number of pairs. But we basically rank them. We rank those predicted contexts based on their probability. So higher probability, more likely there are true contexts, right? So then that's why we select the top. In the field, people try to select a top number of their contexts based on the sequence length. So top L by 5, that means select the top L by 5 number of predicted contexts based on their probability. Then you can calculate how many of them, what proportion of these top L by 5 contexts is correct. That is precision. We can also do the same thing for the top L by 2, because sometimes you need actually more context. Then you check the top L by 2 context predictions. Yeah, pretty much like an image classification. So you classify images into different categories. So in ImageNet classification, they ask you to select 5 labels, right? Ranked by the probability. Right. Then to see whether or not one of them is correct or top one is correct or not. Yeah.
Okay. Thank you very much.